Hey everyone, welcome back to Windows 8 Studies. I hope you all are doing well. So in last class we learned about dosage compensation. Dosage compensation is a phenomena by which differentially numbered sex chromosomes in males and females, they are balanced by equalizing the expression of the genes which are associated to those sex chromosomes. For example, we learned about the X chromosome inactivation in mammals, in which the females who have one extra copy of the X chromosome compared to males, that one extra copy of X chromosome is inactivated. So females, the mammalian females, or the human females, they have two X chromosome, while males, they have one X chromosome and one Y chromosome. So the expression of X-linked genes would be in double amount compared to that of the males, right? So, like in females, the sex chromosome to the autosome ratio. So, they have two X chromosomes, okay, on the diploid set of autosomes, and that is the ratio is 1. While in the males, they have one X chromosomes on a diploid set of autosomes, that is why the ratio is half, which is 0.5. So this is exactly half of the females. Okay, so the females would have double the expression of the X-linked genes compared to the males. So for achieving the balance, the females who have two X copies, the one X chromosome, the one copy of X chromosome is inactivated, and this is done by heterochromatinization. Okay, so the one X chromosome is inactivated, and this phenomena is known as X chromosome inactivation. And by this X chromosome inactivation, the dosage compensation is done. The dosage of the sex chromosome is balanced. Okay, so today we are going to learn about the X chromosome inactivation, the types, the methods by which X chromosome inactivation is done. Okay, let's learn about that in detail. So there are different methods by which this can be achieved. One being the random X chromosome inactivation, and other is imprinted X chromosome inactivation. Okay, so I hope you remember in last class we learned about Lyon's hypothesis. Mary Lyon, she suggested that the X chromosome which is going to be inactivated, that would be random. So the choice of X chromosome will not be pre-decided. Rather, it will be random. So either the maternal copy or the paternal copy of X chromosome can be inactivated. But opposite to that, unlike random X chromosome inactivation, imprinted X chromosome inactivation suggests that the X chromosome which is going to be inactivated, that will be decided by the parent of origin manner. Okay, so that will be parent of origin specific. So it says that the X chromosome inactivation won't be random, but it will be already decided that either the maternal copy of X chromosome will be inactivated or the paternal copy of X chromosome will be inactivated. So that phenomena, that event is not random, but it is parent of origin specific. So for example, the paternal imprinted inactivation is seen in humans. Okay, and this is means it is paternal imprinted, means the copy which is inherited from father, means the paternal copy of X chromosome, this will be inactivated. Okay, always, no matter what in which embryo it is going to be happen, in all of the cases, the X chromosome which is inherited from the father, that will be inactivated. So this is already decided. Such inactivation is known as imprinted X chromosome inactivation. Okay, but in humans we have the random X chromosome inactivation. Then why did I say that humans show paternal imprinted inactivation? Yeah, we'll discuss about that. Again, there's one more thing to the imprinted X chromosome inactivation. The we the paternal imprinting is more common compared to the maternal imprinting. Means the paternal copy of the X chromosome is silenced. This is to ensure that the development of the embryo goes through the maternal experience, because we are assuming that the maternal environment is more beneficial to the fetus, right? And for ensuring that it undergoes the maternal environment, we are not silencing the maternal copy of X chromosome, rather we are silencing paternal copy. This might be a reason behind the paternal imprinting being more common. So maternal imprinting is a rare phenomena. So we generally see the paternal imprinted X chromosome inactivation. Okay. So let's learn about the X chromosome inactivation in humans, because it is a quite interesting phenomena. Okay. In humans, the X chromosome inactivation is random. Okay. But it depends on the developmental stages. So for that, let's learn about development of an embryo first. Okay, here you can see the embryo is being developed from the before fertilization phase. So the sperms, they fertilize egg, and this fertilization creates a single cell known as zygote. The two haploid nucleus fuses, and they form a diploid zygote. Okay, and from this single cell, the development of life starts. So the zygote undergoes cleavage, means it undergoes the division. So it forms two cell stage, four cell stage, eight cell stage, till the cell stage, which is quite important, that we are going to discuss now, is the morula stage. Okay, when the zygote reaches to the morula stage, when the embryo has achieved the morula stage, till the morula stage, okay, from here, from here to till the morula stage, the X chromosome, which is going to be inactivated, that will be imprinted. Okay, so till morula stage, we see imprinted X inactivation. Okay, so the zygote, it has already received one X copy from mother and other X copy from the father, right? So from the zygote to the morula stage, the X chromosome which is going to be inactivated, that will be paternally imprinted X chromosome. Okay, so the paternal copy of X chromosome will be silenced, but this happens till the morula stage. Then comes the blastula stage. When the blastula stage, the blastocyst is formed. When the blastula stage is reached, blastula stage is the only stage where both of the X copies are active. So the X chromosome, which was inactivated till the morula stage, that is reactivated. So the X chromosome, it is reactivated, and this is the only stage where both of the X chromosomes, they are active, and that is why we say that the blastula stage is the only stages in humans where the where the 2x dosage can be tolerated. Okay, means this 2x dosage creates imbalance, right? We have already learned that, but only in the blastula stage, both of the X chromosomes are active, and that is why it tolerates 2x dosage. Okay, there are cells known as primordial germ cells. Also, they can also tolerate 2x dosage. Okay, they also tolerate 2x dosage. Again, so it was seen that blastula is consisted of inner cell mass and the blastocyst has inner cell mass and the trophoblast. So in the blastula stage, we have embryonic stem cells, right? So these embryonic stem cells, they have 2x dosage. Okay, so those embryonic stem cells, they are pluripotent. So it was seen that pluripotency is somehow related to 2x dosage, because again primordial germ cells, they can also tolerate 2x dosage, and the blastula stage, the embryonic stem cells, which are pluripotent stem cells, they can also tolerate the 2x chromosome dosage. So it was seen that these both of them, pluripotency and the both of the X being active, this is somehow related. We will also learn about this and we will discuss about this in detail, but this is not the topic of focus for today. So the take home message for you is that the blastula is the only stage where 2x dosage can be tolerated. So the inner cell mass, okay, this inner cell mass, which is pluripotent, it is going to be have both of the X copies active, but this the trophoblast, or the trophoectoderm, which is going to form placenta in future. This trophoectoderm or trophoblast, this will show imprinted X chromosome inactivation. Okay, so what happens is that let's do a recap. From the zygote to the morula stage, there will be imprinted X chromosome inactivation. Okay, and then this continues, but the inner cell mass when the morula stage goes to the blastocyst stage, it has inner cell mass as well as the trophoectoblast. So the inner cell mass, it shows the reactivation of the inactivated X chromosome. So it has both of the X copies active, while the trophoectoblast still remains paternally imprinted. Okay, so the paternal copy of X chromosome will be silenced, so it shows imprinted X chromosome inactivation. But the inner cell mass have both of the X copies active. But when this blastocyst, this is further developed, and this reaches to the epiblast stage. Okay, and which then undergoes gastrulation. So from when the gastrulation starts, or we can say from the epiblast cell, there will be random X chromosome inactivation. Okay, so we can say that the proper embryo in humans, it shows random X chromosome inactivation. Okay, so we can conclude that humans show random X chromosome inactivation except for the placenta or trophoectoderm, as well as the embryo till the morula stage. Till the morula stage. Okay, so just to summarize, the imprinted X chromosome inactivation, it is seen in marsupials, the primitive mammals. Okay, the marsupials have imprinted X chromosome inactivation only in all of the developmental stages. Then it is also seen in the humans, but only till the morula stage. Till here only. Okay, and then it is also seen as trophoectoderm or the placenta. The trophoectoderm will develop and it will form placenta in future. But the random X chromosome inactivation, which is a widespread phenomenon, this is seen in humans in the embryo proper from the epiblast stage when the gastrulation starts. Okay, so this is it about the different methods of the X chromosome inactivation. In next class, we will learn about the different stages of X chromosome inactivation, means the different stages, different steps by which the X chromosome is inactivated. So we will learn about that in detail, step by step. We will also learn about X chromosome inactivation center. We will also learn about how the choice of which X chromosome is going to inactivate it. That is random, but again, that is also an important factor how it is done. Then we will learn about the establishment of X chromosome inactivation and how this X chromosome inactivation is maintained throughout the life of an individual. So we will learn in detail and we will discuss in great details in next class. But today's class is done with this. In next class, we will also learn about XIST long non-coding RNA T6 RNA. Till then, see you. Bye bye.